I think we're all set up and ready to go. I'm happy to introduce Dr. Maksim Plikus. He's at the University of California at Irvine. In the, what department are you in? I'm forgetting.
I'm in developmental and cell biology.
That's right. So Max got his start as a graduate student at was at USC, and then he moved on to do a couple of postdocs, one at USC and then as well as the University of Pennsylvania, and then finally moved back to California to UC Irvine in the Department of Developmental and Cell Biology in 2012, and he's been rising through the ranks there, and somewhat recently was promoted to full professor. He does some actually really cool stuff with hair regeneration, and hair regeneration is really nice because it's people use it as a hair follicle, or use the hair follicle and the stem cells as kind of a mini model for regenerative medicine. And he's been doing some really cool stuff in this world, and so I figured that he would be a good person to invite and have a talk to us about this really cool stuff. So take it away.
Yeah, thank you very much, Scott. To add to your introduction, my PhD was actually in the pathology department, so I actually have a PhD in pathology. Okay, so yeah, let's briefly talk about what hair is. I think hair itself does not require introduction. I think Scott correctly pointed out, a lot of people made careers like presenting hair follicle is a very good model for studying regenerative abilities of tissues, just because there's very many stem cells in each hair follicle. It constantly goes through growth cycles. You can call them regenerative cycles. They're on the skin surface, and they very accessible for studies, including some fairly sophisticated stuff like light imaging, follicles. But if you sort of go back to the basics and ask, is one hair enough? Is understanding how one hair work enough to know what hair is? And the answer probably is not. So when I think of hair, this is what I think. There has to be hundreds of thousands of them, and they need to all work together to produce fur in most of the mammals. And having thick fur is a highly adaptive feature. So to fully understand what hair is and how it functions, we need to understand how they work together as a very large population to be able to produce very versatile fur that we observe across mammals. Now, when did hair first came about? It's kind of shrouded in the mystery of, it's believed to date back as late Triassic period. So we're talking over 200 million years ago. So there's a little map of paleontological periods, and shown on the right is artist representation of what possibly mid-Jurassic looked like. Lots of dinosaurs of all kinds, but between them we're running this mammals, small little mammals. And it's well known that they were already really furry then. So they're not really truly mammals, but they're what's called mammaliform. So, and these predecessors of modern day mammals are no longer around. And here's a really cool example. So, this is mid-Jurassic. It's an animal that looks a little bit like a beaver, and there's a really good imprint of its fur. And, on analysis, you can definitely say that they had a lot of hair, and the hair had multiple types. So it was already sophisticated then. So what we have now is a result of a very, long evolutionary process. So from evolutionary point of view, what's the biggest novelty about hair compared to other derivatives of skin that existed in earlier ancestors? And the way I think of it, the key evolutionary novelty is invention of a growth cycle. It actually evolved twice, one in mammalian ancestors for the hair follicle, and second time in dinosaurs for feather follicles, and what now are feather follicles of birds. So what this implies is that a small tissue unit that is rich in stem cells can go through recurrent cycles of coordinated growth, and in each cycle, a functional output is produced. In the case of hair follicle, it's a hair shaft. So every time follicle makes a new hair shaft. And hair has various characteristics like length, color, bands, and this is all very important, but at some point there's an end to it. So the growth phase has an end, and then cycle repeats itself. This contrasts, for example, teeth. So teeth, in many ways, developmentally share characteristics with hair follicles. They also have ectodermal appendages. But in order to make new tooth, you have to start from the beginning of morphogenesis all over again. So you need to repeat the whole morphogenetic cycle. You make a tooth, the terminal structure. It doesn't have about ability to recycle itself, while hair follicles can. And what's very interesting is that between individual cycles, same follicle could actually dramatically alter the hair shafts that it's putting out. The length could change, the texture and the color. And this is just demonstrated here with one example. So this is wild boar. These kinds of examples are very prevalent, that adult form of the same species and the newborn form fur differs dramatically, like including color pattern, length, and texture. So, many baby animals have very fuzzy, cute fur, not so much for adults, and color changes. And this is just one example. What this means is that there's a mechanism that could reset the program for the properties of hair. So with that in mind, and the way I kind of think what is a good analogy for hair follicle is it's kind of a nature's 3D printer. So analogy is following, like 3D printers, they could print three-dimensional structures many times. There's an on button, and there's some molecular on button at the beginning of cycle in the hair follicle. That's what we're really curious about. But to sustain this numerous print cycles, printer needs to have ink of some form. So hair follicles ink would be its long-lasting epithelial and melanocyte stem cells that reside in a specialized structure at the upper part of the follicle. It's called bulge. So these long-term self-renewing epithelial stem cells and melanocyte stem cells are housed in the same compartment. And this compartment is sort of removed from the active print unit, which is at the base of the follicle. So printing of a hair occurs through coordinated processes of cell proliferation and terminal differentiation within the epithelial lineage. Now, print process needs to be controlled by some kind of program. And we can load a print program to your printer, and we could make in 3D printer various shapes between cycles. So where is, so to speak, CPU within the follicle? So I think a lot of information about the properties of the hair are actually contained within the mesenchymal compartment. So it's a mesenchymal niche cells that form this tight little cluster at the base of the follicle. And they are a specialized type of fibroblast called dermal papilla cells. So a lot of interest in the field is to better understand these mesenchymal drivers of hair growth, because they're vital. Their removal leads to complete shutdown of hair growth. Now, another very interesting aspect of this idea is that, if you remember that picture of a seal, it's not just about one follicle printing effectively a hair strand. It's about all of them working together. So this is an inverted view of a newborn mouse skin, and you can appreciate here. So what skin really is this two-dimensional array of like densely positioned 3D printers, the hair follicles, that all are working relatively simultaneously to output what will become fur. So in that sense, it's like in mega factory that produces, processors. So there's an array of this lithography machines in this factory floor. So there's an array of 3D printers in the skin, and there needs to be coordination between them. So how do animals achieve coordination for effective fur production? So there's actually more than one way. Some animals are strictly seasonal, meaning that they go through either one very coordinated or two coordinated cycles. So many Arctic animals could produce two sets of fur, and there's snow white fur for the winter, and there's always brownish fur for the summertime. And that's regulated by the yearly cycle of the systemic hormones. But many animals that are non-Arctic do not have this top-down approach for controlling hair growth. In fact, what they have is bottom-up approach. So without systemic control. So if you were to actually take a typical wild-type mouse and you to shave it, some of you might have done it. Depending on age, you would see something like this. There are clear patterns of pigmentation on a skin surface. Now, epidermis in mice is not pigmented in the dorsum. So when you see this pigmentation, it comes from actively growing hair follicles, because melanogenesis is contained to the print center, the bottom of the follicle, and you see it through the skin. So, and gradations in pigment sort of kind of tell you the stage of maturity of the follicle. So the darkest spot is shown here, called the initiation center. That's where the most mature follicles are that are growing. They've been growing the longest. And then what you see is that there's this gradation wave of pigment spreading in all directions. I highlighted one going toward the tail. And that's the spreading wave. So meaning that follicles across this vector, they will be progressively younger in the growth phase. And what's very interesting, you will often find areas that look completely pink. That indicates that there is no active hair growth. And often the growing and non-growing areas form really sharp boundaries. So that means that skin can partition itself into growing domains and non-growing domains, and growth can propagate between the follicles through a wave-like process. So this wave-like process and boundary is demonstrated here. So now you're looking at the inverted piece of skin in an adult mouse, and hair growth wave is propagating from right to left. So particularly some parts of this hair wave are magnified for you to see, and pinkish color that you see, this is actually stain for oil. So it's a lipid stain, oil redo. It just highlights position of the sebaceous gland. So each follicle has a gland, and it was done just to actually for you to see that even in the areas that are not growing, where it says T stands for telogen, the rest phase, there's lots of follicles that just dormant. And here you can see progressively younger follicles as you move along the wave, and you get to a point where you just barely start seeing pigmentation. That means that these follicles have entered hair growth and just initiated melanogenesis, and they exist very close to the wave front. So if you were to look at actual wave front, so which is just a little bit more to the left of the pigment wave front, you will start seeing molecular events of this wave propagation. And here's an example. So we know in the field that hair growth is highly dependent on canonical wind signaling. And now you're looking from left to right, it's a Holman piece of skin showing the one of the canonical wind reporters, bad gal. And numbers 1, 2, 3 represent individual hair follicles. And follicle number 3 would be like the youngest growing follicle. It's a very, very close to wavefront. So if you were to micro dissect these follicles and look from the side, where is the location of the wind activity? You would notice differences. So I will not talk about follicle number 1 because it's clearly in a completely full growth phase, and it has its own wind activity. But what's very interesting is follicle 2 and 3. The one that's the youngest has wind activity that is restricted only to the mesenchymal niche cells, to the specialized fibroblasts. And epithelial cells, which are right above it, HG stands for heterogerm. It's like derivatives of the bulge. It's yet another essential epithelial population of cells for hair growth. So the orange HG, it's negative. But the follicle number 2 is a little bit more advanced because it's further away to the left along the wave. It actually has wind activity both in the mesenchymal and epithelial compartment. And this is what this means. So as follicles sort of progress toward the actual event of hair growth initiation. So the follicle that I'm just showing you here on the very right is about to grow. And green represent Wnt activity. So it's mesenchymal niche, so the dermal papilla is Wnt active, and the epithelial hair germ cells are Wnt active. And transcriptional changes occur in this mesenchymal Wnt active niche through a couple additional signaling pathways, including FGF7/10 signaling and TGF beta pathway that amplify need for growth to epithelial cells. So epithelial cells actually are getting both the Wnt and FGF and TGF beta signaling all synergizing, telling them, we really think you should grow. Signal is very strong. But prior to that, the first event is that of only the mesenchymal niche. And that Wnt signal appears to propagate from a growing follicle that is high source of Wnt production to this rest follicle. And it hits the mesenchymal niche first. So it sort of puts follicle into an alert state, but it's not yet enough to grow. Along the way, Wnt signal comes to each dormant follicle from many neighbors. So then the strength is very high. So along the way, everybody gets engaged. So every single follicle will grow. You could also probably imagine that to start wave in the first place, you need some kind of spontaneous activation. So I think a very good analogy, actually, and computationally, same model could simulate how hair growth occurs and how fires spread in the forest. So, forest fires, they spread as a wave from the burning tree to the non-burning tree. But what starts the fire is often like initiation sites, small fires, arsons, as an example. So what is sort of the small fires that spontaneously start in skin to actually initiate waves? That involves spontaneous Wnt activation within small groups of resting follicles. And sometimes we could actually observe them. So at any given time, close to 5% of dormant follicles in the resting skin would go into Wnt active state. We don't know how long they persist there, but for a period of time. But that solitary Wnt activation event is actually not enough to engage this follicle into growth. So they probably reverse back out of it. So it's sort of stochastic. But with some low probability, a group of 5 and more follicles in close vicinity can go into Wnt active state. So the secondary messages now become jointly amplified. And we believe that such events lead to initiation of actual hair growth. So, this would be a small forest fire, and then through wave propagation mechanism, which is substantially more efficient and has much higher levels of growth activating signals, it will spread through the neighborhood. And for example, in mice that overexpress canonical Wnt signaling, this threshold for spontaneous initiation drops dramatically. So in the skin of mice carrying epithelial overexpression of Wnt7a, we see very dramatic activation of hair growth. It becomes quite abnormal. Just hairs could actually even grow by themselves. Now they don't even need neighbors. Spontaneous initiation sites form all over the skin. So this is conductin like Z, so it's another Wnt reporter. So in this mouse, every single mesenchymal niche of every single follicle is Wnt active, and you have solitary hairs growing to growth. You have small groups of hairs occurring everywhere at a much, much faster pace. So these mice show dramatically excessive hair growth as the result of it. The same principle, so formation of rare spontaneous hair growth initiation sites and very effective wave spreading of hair growth through the rest of the neighborhood, also operates in other animals. Other rodents such as rats display them, but also the non-rodent species. And here's an example of a rabbit. So this is a rabbit that was shaved. Normally would be covered by fur, but if you were to shave it, the same principle applies, that pigmentation is restricted only to the actively growing hairs. So it really looks like a bit of a fractal. So you have, if you kind of follow where I'm pointing, you have spontaneous initiation sites. So there's this little tiny, pigment sites. These are spontaneous initiation sites. Then you have this expanding sites that spread signal through a neighborhood, and actually small waves spread from them. And when they come close to one another, they coalesce and they produce this fractal-like boundaries that continues to propagate. To really appreciate how dynamic it is, what I'm going to show you next is sort of like the photoshopped version of the hair growth patterns in this same rabbit that preceded what you're looking at now. So rabbits, we shave them every week, and then this is how we'll look. So I'm going to make it darker. So here, a month ago, the rabbit that you just saw, the only hair growth site that it was just at the tail. So there was just one domain and the wave was propagating. And you can appreciate that it's propagating because 10 days later, it expanded to the yellow boundary. Yet another 7 days later, it propagated even further. But in other events that start occurring, the spontaneous initiation site. So you see a couple yellow spots, they occur, and then with time they propagate and then they merge. So that's the pattern that emerges. So therefore, many mammals have a self-organized mechanism for producing fur that does not rely on any central control, such as central hormonal control, but relies on the crosstalk between adjacent hair follicles and scaling up of this behavior. And it could easily scale from a small body of a mouse to a much larger body of a rabbit, as an example. But in the end of the day, when follicle makes decision to go from rest to growth, be that inside of the spontaneous initiation site or at the front of the sweeping wave, the decision is always individual. Although there's a collective behavior, so many follicles make same decision when signal is high. In each follicle, decision is individual. And we believe this decision is made by the CPU cells that constantly read out signaling input that comes to them from outside of the follicle, what we call long-range signaling inputs that they receive, such as from the neighboring hair follicle. So studying these cells is kind of really important, but historically has been really hard. Just like in general, in skin, we understand much better epithelial cell biology than the mesenchymal cell biology of the skin. And only recently, sort of with the expansion and accessibility of single cell technologies, we can study just really how diverse transcriptionally and now functionally fibroblasts are in the skin, because morphologically they are often indistinguishable from one another. And there are very few unique markers that exist for distinct fibroblast populations that we have identified through sort of a targeted approaches. So dermal papillae fibroblasts, because of their essential role in hair growth, would be very attractive to be able to kind of target transgenically and study. And there are really few tools in the field over the last 2 decades. So recently we came upon a fact that leptin receptor is expressed in a very restricted pattern in mouse skin, and it's primarily localizes to these dermal papillae cells. So in mice that have been produced over a decade ago, where constitutive Cre is driven by the alternatively spliced isoform of leptin receptor called LEPRB isoform. It's actually a minor isoform that represents only about 7% of the whole transcript of this gene. This Cre becomes very efficient in the dermal papillae cells. So you can see it on the whole mount. It's really in every single dermal papillae, and you can see it on histology. There's some other minor expression sites like endothelial, rare endothelial cells, but it's primarily in the cell. So it got us really excited. The fact that this Cre is driven by smaller isoform of the gene also delays the onset of Cre activation. In a sense, it's artificial because the transcript for LEPRB, all isoforms transcript, becomes active very early. Already at birth, it saturates in all dermal papilla fibroblasts. But Cre activity, as judged by the reporter, saturates slowly over time and reaches saturation only at about a month's age, which is good and the bad news. Bad news is that you cannot use this Cree line to really study late stage morphogenesis of the hair, but it actually sort of effectively skips morphogenesis. And then if you perform gene overexpression or deletion using this Cree, you would primarily study the effects of these genetic perturbations on a full adult hair follicle. So we kind of acknowledge that fact, and excited by this accidental finding, we actually went on to create a new Cree line. Genetically was constructed the same as the constitutive Cree, but here we have Cree R version of it that's also is driven by that minor isoform of leptin receptor. And it becomes very, actually, as you can see here by the tomato reporters, very efficient and robust in the dermal papillae at 3 different induction time points that we tested thus far. So I think it will be a very useful genetic tool for studying the cell. Now, to demonstrate that it is actually useful, we sort of chose a low-hanging fruit. We overexpressed a dominant active form of a hedgehog pathway gene called smoothant. And the idea was that we knew that hedgehog is essential for fibroblast biology. It's an important lineage regulator, including in dermal papillae cells. So we anticipated a phenotype, and we received a phenotype in this mice, but it had late onset. So till about day 50 of life, mutant mice overexpressing dominant active smoothing don't look different from controls. But after that, they start to look very different. So I already mentioned to you in the beginning, if you were to shave mouse, depending on the phase, you would often actually see just complete areas of resting follicles. No follicles are growing. So for instance, the top mouse, if you were to shave it at about day 50 till day 86, they will not grow a single hair because everybody is coordinated in the rest phase. But the mutant mouse will start sprouting this sparse hair. It looks completely not normal. There are no waves anymore, so waves are gone. And on histology, you'll see it's kind of the salt and pepper patterns, like hairs grow here, not the neighbors don't grow. Completely different, and it sort of could be appreciated here. So these mice show this salt and pepper pattern. It's almost autonomous hair growth. Now, with high activity of hedgehog inside of the dermal papilla cells, follicles do not require extraneous signal from neighbors, so they don't need that wave, so they could just grow on their own. And that's quite different from control mouse. So this really told us that whatever genetic perturbation that we introduced to dermal papillae has to put them in sort of this superactive state from a signaling point of view, where they produce large numbers of the downstream molecules that are essential for inducing hair growth. So this presents an opportunity to study what are the downstream molecules that actually initiate hair growth by signaling to epithelial cells to tell them enough signal has been received. We've been in a really active state. There is definite good biological reason to go into a growth phase. Of course, here in transgenic system, we fooled it, right? We fooled it by smoothing overexpression. So to study that, we performed single-cell RNA-seq experiment, but we chose to sequence whole skin of mutant and controlled mice just a few days before the morphological phenotype. And the logic was that if you do it when phenotype appears, you will be actually studying transcriptome of the consequence of the phenotype, not the cause of it. So we wanted to test it a little bit earlier. So this was, I believe, on day 47 of age. So when mutant control mice look indistinguishable. On single cell sequencing, what many of us do now, we then annotate cells and we perform joint clustering, and all cells look the same, with a little bit of an exception, and they're highlighted in this dark circles. So mutant mice, so the Tisney plot is shown on the right, and mutant data is on the very right. So you see these 2 clusters, the salad green and sort of this navy blue kind of color. These clusters are almost exclusively unique to the mutant, and they don't exist in control. They are both fibroblasts. They are both Cree expressing fibroblast cells. I will mostly talk about the salad green because they are the dermal papillae cells that are now transgenically activated. So the rest of the salad green cells, these are actually just normal dermal papilla cells that also exist in control. But the cluster here inside of the circle is essentially mutant unique cluster. So we'll not go in too much depth about analysis of the clustering, but we were primarily interested in signaling, because whatever the downstream changes, downstream of hedgehog overactivation in this group of cells must be driving precocious hair growth. And the way to study that in single cell data is with tools dedicated to analyzing signaling networks. So recently, a year ago, we, with the mathematicians here at UCI, actually kind of developed this tool that we call cell chat. So what it does, it could take any single cell data as an input and then cross-references gene expression to manually annotated database of all known signaling molecules across all paracrine and orocrine and cell-cell contact signaling pathways. And then after doing that, it performs bioinformatic analysis. And the output that it produces are inferences on signaling communication. It becomes actually quite useful. So it does a lot of this manual job for you within minutes. And then what it gives you is a ranking of significantly changed signaling pathways between two data sets, such as control and mutant single cell data. It's quite commonly used right now by others. So I will just show you an example of what we did. So here we actually pre-selected to run what we call cell chat analysis on signaling communication only between cells that we know exist in close physical proximity, thus they could communicate. So we chose the dermal papillae cells, we chose the hairoderm epithelial cells, and the bulge epithelial cells. So they're really within the morphogen gradient from one another. And when we did that, software produces essentially two-dimensional reduction plot, and it's shown here in the center. But each data point on this plot is actually not a transcriptome of a cell, but it's rather the structure of a signaling pathway network, let's just say Wnt signaling pathway network. And then mutant and control are put together. And actually the signaling pathways intuitively also cluster based on the structure similarity. We actually, in this particular case, got 6 clusters of signaling pathways, and they are sort of magnified. So each cluster is magnified, and they are annotated. So what I can tell you is that many signaling pathways mediating communication between these mesenchymal epithelial cells and follicles don't change between control and mutant. Here's an example: canonical Wnt signaling. It's really very, very similar. They're in the same cluster. FGF signaling, very similar, and on and on. But certain pathways dramatically change. Today, I will just tell you about one. So TGF-beta pathway prominently changes. So in control, which is the colored circle, it's in cluster 1, but in the mutant, it moves to cluster 5. That immediately indicates there's a prominent change in this pathway. So then what we can do is, after looking at the software predicted ranking of changes, we can then zoom in into the pathways to see, well, what's behind that, what's the difference. And this is what you will get. It's, you can call it like a hairball diagram. So software then could arrange all clusters along the periphery of the circle. They colored, and the sizes are reflective of the number of cells in this cluster. So the larger the diameter of the individual circle, the more cells there. Then lines between circles, the periphery of the plot, are colored according to the source. Okay, so what this means is that this dermal papillae cluster 2 is signaling to hairoderm cluster 2 through, and that's by the color. Okay, and then dermal papilla cluster 1 is doing the same. It's signaling to cluster, hairoderm cluster 2. So that's how you read it. And these little loops that go on itself, that means that there's an autocrine signaling. So when cells signal likely to themselves, again, it's all predicted. So if you were to look at the control and the mutant TGF-beta signaling pathway network, just visually, you could appreciate, yeah, it's different and it's increased. So there's more signaling. The pink and light purple cells, which here we annotated as DP, dermal papilla 6 and 7, these are mutant, essentially almost mutant-specific states. And they, you could appreciate here, become really dominant sources of TGF-beta signaling. Then Another very interesting detail, while in control, the dominant ligand driving this communication are classic TGF-beta ligands, primarily TGF-beta 2, and next ranked would be TGF-beta 1. In mutants, TGF-betas, the classic ones, are overpowered by a signaling molecule called SCOOB3. So SCOOB3 or SCOOB3 belongs to a family of signaling molecules with 3 members, 1, 2, 3, and ligand 3 has TGF-beta activating properties. And on here on RNA scope, it becomes overexpressed in germinal papillae cells of the mutant at the time of sequencing, so the same stage time point. So what do we know about TGF-beta and hair growth and SCOOB3? So this is somewhat summarized here. This is primary literature. So on the left is a little bit of the molecular knowledge on SCOOB. So they are initially produced as transmembrane proteins on target cells. They tethered to membrane, but then they undergo proteolytic cleavage, and then they generate soluble peptides. So this cube-containing peptide, shown here, could act just the classic TGF-beta ligands on the TGF-beta receptors in a paracrine fashion. So it would activate canonical TGF-beta signaling through SMAD2/3 phosphorylation. Now, what we know from works from Elaine Fuchs's lab about a decade ago, that during normal physiological activation of hair growth, when follicles naturally transition to growth phase, TGF-beta classic ligand 2 becomes upregulated in the dermal papillae cells, and it signals through a paracrine mechanism to the adjacent epithelial cells. And this signaling then is really important for activating them, putting them into proliferative stage. So clearly SCOOB3 hijacks this essential pathway. And by transgenically producing high quantities of this TGF-beta ligand, we short circuit this such that follicles are now much easier activatable. Another interesting thing that we realized that SCOOB3 is really not some kind of like an artificial molecule that normally should never ever be present in the cells. In fact, it's an endogenous TGF-beta activating molecule in the hair follicle, but in a cyclic fashion. So this is RNA scope for SCOOB3 across different postnatal time points, and it clearly shows this on and off pattern when expression is restricted to dermal papillae of only actively growing follicles. So it doesn't activate at the onset of hair growth, but it comes a little bit later, and it probably becomes an important TGF-beta sustaining ligand from these niche cells. So I think in our transgenic system, we precautiously activated in these cells, and that short circuits the system. The reason this is sort of summarized here. So we believe that by transgenic perturbation in hedgehog, downstream of it is SCOOB3 that basically short circuits this more orderly TGF-beta 2 dependent activation process and basically overpowers it. And the way we're also kind of convinced by that in this model is that just the microinjection of the recombinant SCOOB3 protein into the skin of mice on the agarose beads could induce precocious hair growth at the site of the beads. Well, our control beads, which we typically soak with Bowen serum albumin, are neutral in that sense, so they're not activating. We know it's TGF-beta pathway dependent from some of this immunohistochemical data showing that mutant hair follicles, even before visible growth, they have precautiously activated TGF-beta signaling based on phospho-SMAD2 staining, and it sort of corresponds to high expressions of SCOOB3, but also just really topical application of TGF-beta inhibitor onto the skin of mutant mice nearly completely rescues this salt and pepper precocious hair growth phenotype. So it blocks it, and we think it's the lead pathway. So I guess with this data, I just wanted to highlight the fact that now we have genetic means to target dermal papillae cells and it becomes kind of really useful system in which we could start asking functional questions. So what is downstream of the Wnt activation in dermal papillae, and possibly what's upstream of it? So, and this story that I shared with you with Scoop 3 is story showing that we can identify novel downstream components that have not been previously implicated in hair growth. But going back to that example of a seal, again, hair growth is really not about individual follicles, but it's about all of the follicles together. And what I showed you also that in adult mice, this process is self-organized. It's bottom-up approach. When follicles communicate through paracrine signaling, they integrate individually incoming signals, and then they make decision to grow or not. Within the spreading wave, decision is almost unanimously to grow. Otherwise, it has stochastic probability, such as when initiation centers form. But think of a newborn mouse. So newborn mouse is, so when house mouse is born, they slightly underdeveloped compared to some other rodent species. So they lack fur. So they're born without fur. And of course, thermal protection is not as good as an adult furry version of a mouse. So soon after birth, what I think big job for skin of a mouse is to grow as much fur as possible. So I don't actually know the total number of hair follicles that mouse skin has. It's probably on the order of several 100,000. Let's just assume it's 100,000. So after one cycle, the fur density would be 100 hair filaments. The mouse continues to grow, but new follicles are not added. So that means that 100,000 filaments become spread. So fur actually with age becomes a little bit thinner. And one really interesting aspect of the way mammalian follicles manage their fur is that they don't shed the old hair after they grew the hair, they actually retain it. So they glue it inside of the follicle. So when mouse gets bigger, but the hair density is decreasing, one way to solve it is to very quickly grow second set of fur, so that now each follicle will have 2 strands of hair glued inside of it. So your hair density would be now double the number of follicles, it would be 200,000. And then very quickly go into the third cycle. So you bring it up to 300,000. So you can get really dense fur by rapidly going through cycles. And that's what young mice do. They achieve approximately 3 hair strands per follicle very quickly, and then they dramatically slow down, because after that the fur density would be considered pretty high. So how is this very rapid postnatal growth waves is achieved? Through the self-organized mechanism, because I mentioned to you, it sort of relies on this rather really inefficient spontaneous initiation events that could take weeks to occur. So that would not be okay in young mice. So one really interesting and very classic observation done in rats already, in 1949, is that in young animals, if you were to skin them or just look from the top and you draw a midline, the hair growth patterns are almost mirror images of one another on left and right. That is clearly not stochastic. This doesn't look like the rabbit that I showed you. It doesn't look like fractal. It looks like a complete bilateral symmetry. And on the right, I show you examples of young mice with like red lines sort of put through the middle, also showing that there is this kind of bilateral symmetry, often very symmetric. So how can this bilateral symmetry achieved? Through model simulations that we performed, the only condition that could produce bilateral hair growth pattern requires an assumption that skin is actually not flat, how we sometimes like to think. We look at the animal from the top thinking it's sort of a bit of a rectangle, but it's not. If anything, it's more of it's more of a cylinder, because there's also skin from all around. So the whole body of the mouse, it's really a balloon or a cylinder. So you have to assume that hairs form waves across the entire cylinder. And the second assumption is that hairs in the ventrum somehow have to have endogenously higher probability for initiation. And in this simulation, and kind of the simulation is shown at the bottom, green is actively growing simulated hairs, red is inactive. You would observe bilateral pattern forming due to the faster growing ventral hairs producing bilateral waves that go through the shoulder regions and essentially enter dorsum from both sides. So it's an artifact of like us looking at mice from the top, but it's really a bilateral wave spreading from ventral to dorsum. We actually went on to validate that in young mice, this in fact exists. So that ventral hair growth starts always faster than dorsal, and then there's a bilateral wave that propagates. So shown on the left are luminescent images from the wind reporter mouse. There's two red boxes that are put around day 24. We'll just kind of focus on that. So that same mouse shown from the top and the bottom. And when you see luminescence, that means that hair growth has already started. So day 24, hair growth already started in the chin and the belly area of the mouse, but nothing in the dorsum. If you fast forward to day 26, you will see that that hair growth now has progressed dramatically across the ventrum, but it also is entering dorsum as a wave. So you can see bilateral wave as day 26 coming around the shoulders and it's going down. And graph on the right shows temporal advancement of ventral over dorsal hair growth. And the colored blue area shows that in the 3 consecutive cycles, ventral hair growth is always faster than dorsal. And that drives this fast recurrent hair growth waves that eventually lead to triple the hair density to the number of follicles that the mouse has. So what makes ventral hair follicles so much faster? It is, we explored that transcriptionally. So we performed whole skin. This was, pre-single cell days. So we performed whole skin RNA-seq at the same time point, comparing skin in the chin of a mouse, in the center of the belly, and the dorsum. And at the morphologically identical phases of the growth cycle, in this case, I'm showing the rest phase. And what you can see is that they cluster by site. That means there's substantial transcriptional differences in the whole skin, even though morphologically it looks identical across the 3 areas. So when we focused on the signaling genes, it became pretty clear to us that major differences are in 2 key pathways that are regulating hair growth: the activating Wnt pathway and the inhibitory bone morphogenetic signaling protein pathway, BMP pathway. Here, what I'm showing you is the whole mount Wnt activity data from same mice. At the top is the Wnt reporter, at the bottom is BMP reporter. So Wnt reporter activity, the important part is where the mesenchymal niche cells are. So I will put red dots over it so it will be easier for you to see. So you can see they really differ. So in the dorsal skin, there's very little Wnt activity in the mesenchymal cells, but all of the epithelial stem cells have high BMP activity, which is highly inhibitory to hair growth. So this is resting follicles. They're really not ready to grow. At the same exact time, same mouse, in the chin and the ventrum, pattern is different. BMP is disappearing, Wnt becomes almost 100%. So that means that these follicles are about to grow, and they will be the dominant site for wave initiation. So they will start the wave. And this chin ventral to dorsal wave propagation is what will drive fast fur growth in young mice and will result in this bilateral symmetry type of pattern. Eventually, in adult mice, skin reaches the point where follicles have enough hair strands sticking out of them that they don't need to grow another one. So there has to be another mechanism in adult animals that would essentially prevent excessive hair growth. That would be unnecessary. It would be potentially quite wasteful for resources. So how do you do that? And I want to go back to this example of this mouse that I started my talk with. So clearly wave is propagating from this initiation site and mid-dorsum all throughout the skin, but it's avoiding this coin-shaped region in the lower dorsum. So what is so distinct about this region? Now, this mouse, we've traced its hair growth patterns prior to this picture. And what we knew is that this coin-shaped area just finished its own hair growth a few days before that, meaning that if we were not to trim the mice, it will have enough hairs coming out of each follicle. So there will be no biological reasons for them to grow yet again so quickly. So they are protected from excessive hair growth. And this phase lasts approximately, this protected time, protected rest, vacation, if you want to call it. We call it refractory telogen. And in mice, it's approximately 30 days. So what's very interesting is that this refractory state is dependent on very high levels of BMP signaling to the epithelial stem cells. But the source of BMP ligand is extrafollicular. It's not inside of the follicle. In fact, it's localized inside of the dermal adipocytes, which are adjacent carpet-like structure in the skin. So dermal adipocytes produce high levels of BMP ligand too. And we have hypothesized that this adipose to hair follicle signaling is what maintains follicles in the refractory phase. That's what assures that they are on vacation for at least a month. And by modulating BMP signaling, we could actually put them from competent phase to refractory phase again by just placing BMP soaked beads ahead of the spreading wave. So wave would actually avoid that region. There'll be several dozens of follicles right next to beads that would become refractory again. So it's not an intrinsic property of follicles. They're not able to grow. It's actually an extrinsic effect on these follicles by BMP ligands, either from adipose tissue or beads. So it's very interesting. Mice have one body side where follicles are on a very long vacation. They almost never grow, and it's ear skin. So ear skin in mice is actually fairly densely populated by follicles. So this is looking at the P7 section through the ear. So that's when follicles just completed morphogenesis and they're growing. And that's the first time they're growing. They will produce a short little hair. And there's a lot of follicles. What's very interesting, there is a cartilage. So ear cartilage, shown here by SOX9 staining, exists in very close proximity to hair follicles in this body side. And there are no cartilages close to follicles anywhere else on the body. What's very interesting, if you focus on what I'm showing at the top, so this is kind of like a street light color. So green is grow, yellow shutting down, and red, no growth, stop. So if you were to kind of track the timing of 3 consecutive hair growth in mice in the dorsum, over the first 90 days, they go through 3 growth cycles. So each follicle in the end of the 3 months of life will have 3 hairs sticking out of them. But in the ear, follicles go through 1 cycle, and then they go into the very long rest phase. So they don't grow again for a very, very long time. So they'll have only 1 hair sticking out. So the hair density on the ear is much less because of that. We asked if these follicles are maybe somehow completely defective. Maybe there's something developmentally wrong with them where they're just completely unable to cycle, unlike dorsum. But that's not the case. For example, by plucking hairs or by treating them with high levels of hedgehog activator called SAG, we could actually force them to grow. They will do so, not very efficiently, but they are absolutely capable of growing. So that means that there's nothing wrong with the follicle per se. They're completely competent of going through cycles in the ear, but there's something that really prevents them from doing it. And it's likely some form of paracrine mechanism. What we found is that the key reason for that is high levels of inhibitors of hair growth that are produced by the cartilage. So by bulk sequencing on microdissected ear cartilage, compared to ear skin or dorsal skin, we could actually observe that, as summarized here at the bottom, ear cartilage produces really high quantities of inhibitory BMP ligand, BMP5, as well as a number of other inhibitors of hair growth, including soluble Wnt inhibitors, FGF18, which is a really well-known hair growth inhibitor. So ear follicles are resting for such a long time, primarily because of paracrine inhibitory effect of the cartilage. And we could modulate this, for example, in transgenic mice in which soluble activator of soluble antagonist of BMP called noggin is overexpressed, or in the same mice I showed you some slides earlier when canonical Wnt is overexpressed. Now many more ear hair follicles will constantly grow. So we'll get actually mice with furrier looking ears. So this suggests that, as shown in this sort of diagram, that the entire body of the entire skin of the mouse that has hair follicles that form there, they have landscape-like mechanism for maintaining fur. In certain body size, such as ears, in mice, low density of fur is preferred, and that's achieved through a paracrine inhibitory effect from non-hair structures such as cartilage. In other body size, fur density needs to be high, like in dorsum and oventrum of young mice, and then so they implement this pacemaker-like mechanism when intrinsically more activatable ventral hair follicles set up skin-wide bilateral waves of hair growth. That then slows down in adult mice, and follicles enter into more economical, slower pace of hair growth, where there is a mechanism that's dependent on inhibitory signaling from adipose tissue that sort of prevents them from going into a recurrent cycle too quickly. So they are assured of at least 1 month's hair growth, hair rest. So with that said, I also want to briefly mention two more things. So one is the relevance to humans. So clearly, when we go get haircut, we don't look like mice, most of us. So that means that there's no long-lasting bald spots. Like it seems that as if every hair is constantly growing back. And the key reason for that is that in a scalp skin, the growth phase of individual hair follicles is on a scale of 5 to 7 years in a typical adult. That's very long. That means that they don't require coordination of hair growth. Each follicle is in many ways stuck in a very long-lasting hair growth. So they autonomously can be growing. And the probability of close to 90% of follicles growing at any given time, even autonomously in human, is very high. So they don't need that. But there are certain conditions, both physiological and pathological, where coordination becomes enforced. It's probably a bit of like an atavistic feature in a human. So one time we see patterned hair growth is in approximately 4 months of age in babies. And those of you who have kids, young cousins might have noticed that babies have this transient bald spot that appears, and it's about between 3 to 4 months of age, and it goes away. And most parents attribute it to the fact that baby is constantly in the crib and rubbing its hair and it's just falling out. But that's not the cause of it. It could contribute to a fallout. But the key cause for that is that this is the synchronized exit from a fetal hair cycle by these follicles in the occipital region. And after that, they enter very quickly next growth phase. And because next growth phase starts to last years, the synchronization is gone. Now, in adults, synchronization could be achieved upon pathological states. And one such well-known state is an autoimmune form of alopecia called alopecia areata. So it's an autoimmune attack on the hair follicles that does not destroy follicle, but it puts follicle into a very long-lasting rest phase that could last decades. But if this autoimmune mechanism is shut down, such as recently by systemic JAK-STAT inhibitors that have been recently actually FDA approved for alopecia areata, what patients would experience, they would experience a rapid regrowth of all of these hairs that were dormant for decades. But regrowth occurs not stochastic, but it occurs as the pattern. So it's the same patient just sort of traced over time. So you can see how there's actually progressive spreading from the existing hair growth sides into the area. So it sort of closes in from the sides. And that's a telltale sign that there's a wave propagation that takes place here. But a condition had to be created for this wave propagation. And key condition is that you need to have like a uniform field of resting follicles that are able to get engaged in the wave-like behavior. In a typical adult, this condition does not exist. But we could observe it upon resolution of alopecia areata on a treatment. Okay, and the last thing I want to show you is the following. Let's just kind of use example of cats. So animals that have high fur density to begin with ideally need to maintain it. So, but there's extraneous insoles that come at fur, and they could be different in strength. So for example, if cats groom themselves or each other with their tongues, they actually dislodge some number of hair shafts. So they will cause certain follicles to become empty. They will pull out the hair strands. But the number of follicles and the distribution of these empty follicles will be so low that as a whole, fur will be still very much intact. So I think it would not be a very good cause for another cycle. Opposite to that will be, for instance, some kind of more dramatic form of fighting where, if upon biting or scratching, a big chunk of fur will get pulled out. So you would actually have like macroscopically missing fur, and there will be a naked spot in the skin that should grow back. So how does skin differentiate between a few hairs been gone versus, let's just say, a couple hundred hairs been gone? Is there a mechanism for calibrating the extent of fur loss? Turns out there is. So in the mouse experiment, if you were to actually, many people synchronize hair growth by waxing mice. So you would put hot wax on the fur, then wait for it to cool down, and then you would just pull the hair out. So many people do that. This will induce like dramatic hair regrowth. So then the concept was born that, well, hair plucking induces hair to grow back again, which is correct to a point, because if you were to take tweezers and pluck just one hair, it would not grow back. If you pluck two hairs, they will not grow back. If you pluck 5, 10, 20, they will not grow back. And I'm talking about plucking in a tiny spot, so one next to another. If you pluck 50, all of a sudden it will grow, but with a delay. If you pluck 200 every single time, very quickly, the 200 hair spot will grow back. That means that there's a built-in mechanism that a population of hairs somehow could assess the amount of damage. And it ignores low number of plucked hairs, but it responds if it's over 50. So there's some threshold. What I will show you just 3 slides to conclude my talk demonstrating this point. So in the lab of my PhD advisor at USC, somebody followed up on this experiment that I just described to you, where I plucked tight spots of various numbers of hairs, up to 200. So Chi Chang, he performed yet another form of plucking. He always plucked 200 hairs, but he changed the density, the way he plucked. In some cases, he would pluck 200 hairs right next to another. Always they will grow back hair. This is an example at day 30. 200 hairs will always grow. If he keeps plucking and then leaving few hairs unplucked in between, he would get to a point where there's no response. Even though you pluck 200, you spread them out so much that there's no response, so there will be no regrowth. And in between those two outcomes, there is a very interesting outcome when the hair density of plucking is such that everybody will grow in the plucked region. Not only plucked hairs, but everybody that was unplucked in between them. So it's a collective response. And this is well demonstrated here by this sort of summary. So if you kind of focus on the right, so this is the distribution of plucked hairs and the density. There's a clear threshold where density is just enough such that everybody will grow, no matter if you were plucked or not. And this will look like this. So upon plucking of 200 hairs, over 1,000 will grow back. So clearly this indicates non-follicle autonomous mechanism. And the model is very interesting. So this is my last slide. So the model is such that there is actually molecular sensing of hair plucking in the follicle. It comes in terms of changes in epithelial gene expression. So epithelial cells in plucked follicles will start to secrete an inflammatory cytokine, CCL2. It's sort of cartoon summarized here. So only plucked follicles, such as here, will produce CCL2. The target for the cytokine is tissue-resident macrophages that upon activation will swarm into the plucked area and start to produce additional set of pro-inflammatory factors, including TNF-alpha. Now, epithelial cells in hair follicle are intrinsically TNF-alpha responsive, and they will react by growing a hair. But because TNF-alpha is not targeting only plucked hairs, but everybody in the area, upon certain plucking density, macrophage density will be high enough to produce just enough TNF-alpha that will activate collective hair growth of all follicles. And in mice that, for example, have deletion of myeloid cells, this threshold plucking is gone. So they would not respond. So that mechanism really requires macrophages. This was the last slide that I wanted to share with you. I will end here with my summary slide, just to iterate that each follicle behaves as a 3D printer that grows in a cycle, and the cycle is regulated intrinsically by specialized niche fibroblasts. We call them dermal papilla cells. They act as a CPU of this biological printer. This printer collects signaling inputs from multiple external sources: adjacent hair follicles, adipocytes from adjacent dermal adipose tissue, immune cells that patrol skin, and it integrates them into a yes or no decision to either grow or not grow. This integration results in hair growth across large scale to self-organize. And examples of self-organized behavior is low probability spontaneous activation, so from initiation sites, they're stochastic, and then high probability signal spreading. Excessive hair growth is prevented by introducing non-patterned tissue elements such as cartilage sheets or adipose tissue sheets in close proximity to the follicles. And those non-patterned elements produce high quantities of inhibitor that would maintain large populations of adjacent follicles in the long rest phase. And then there's a damage assessment mechanism. So when through an immune-mediated process, hair follicle populations are able to gauge, have enough fur been lost to warrant new growth cycle, or should we ignore it because it wasn't high enough? So thank you for your attention. I have the last slide here, which I'm going to go to. Sorry, the background didn't carry through, but I think QR code is here, and you're welcome to follow the instructions and scan it and do evaluation. So I really appreciate you coming to this Zoom seminar. And if you guys have any questions, let me know or write them in the chat. I can see chat. Thank you.
Thanks, Max. That was amazing. Very, very mind-blowing talk there in terms of how much progress you've made in actually understanding this system as a whole and how the waves are propagated. I wonder if you've thought of things in terms of information theory and how much, how many bits or bytes of information they need to sort of make the decision about it's time to grow. Like you have the data clearly, that it's not 50, 200 is the threshold for, an actual decisive decision, but then there's this sort of probabilistic iffy decision at 50 hairs. Anyway, it's just fascinating to me. I from having looked at people that were doing the Alpha project in the East about making a decision whether or not to mate. They did some things where they were looking at, information transmission, and you're certainly doing that. It's just not formalized in that way. It's
right. I mean, at the threshold, there's clearly, I think at the threshold of, say, plaque density, there's a gray area, right? So certain experiments would result in growth and others wouldn't at the threshold, because it's really, there's some degree of stochasticity at that point. So yeah, but the fact that this populations of hairs could integrate the damage signal, it's very similar to quorum sensing in other systems. So they really, they kind of communicate, but they're so sparse. So the plucked hairs are not able to directly communicate because distances between them often reach close to like 0.5 millimeter, which is like too large for a meaningful and stable morphogen gradient to form. So they need to use additional cell types, in this case, immune cells, as sort of the amplifiers and this mobile cell vectors to actually bridge this gap. So like they could then actually communicate through a means of like intermediate immune cells over distances that are far greater than morphogenetic gradients.
so
that's
I'm sorry,
quite interesting.
0.5 micrometer, so 500 microns is what you said the morphogen gradient.
Well, so morphogen gradient, I think typical morphogen gradient in tissues, this is obviously something that's highly debatable. I think people say that depends on the signaling pathway. It's no more than 100 micron through simple diffusion, and diffusion in true extracellular matrix is never simple. There's a lot.
No, it's gooey.
But it's typically considered to be no more than 100 microns. So in this case, follicles could communicate over distances that are 5 or even more times larger than this kind of like barrier. So
that's kind of what I was getting at, like as if there was
Yeah.
almost a jet stream between the cells, right? There was actually flow. That's fascinating. It
Yeah.
almost suggests that there might be some other contractile function of the cells too, to sort of, you know, it's speculative. It's just fascinating to me because of the biophysics of this, Max. This is just, yeah, I don't want to.
But I think there's some other interesting examples of this sort of the decision in skin. So for example, scarring. You know, there's certain injuries, as we know, that will result in healing by scar formation, and injuries that are physically smaller than that will result in essentially almost a scarless appearance of healing. For this reason, many of you might know, like Fraxel laser or microneedling produces minute, tiny injuries because the needles or the laser beam is small. Even though you put very numerous injuries per skin, each one is so small that skin makes a decision to not form a scar. But as you increase the physical size of individual injuries to a certain threshold, all of a sudden there's a scarring response. And I think there's again some kind of gauging of the damage size. And then based on that information, is it small, is it big, the decision is made to heal through scar formation versus kind of more scarless healing.
Fascinating, Max. People here in Path 520, I know a lot of you might have some good questions. So Max, we also have another seminar that starts at 2:30.
Sure, it started probably already. So I know that I went just slightly over time. So really appreciate your guys' attention and interest. Thank you.
I certainly would like to contact you some more. There's certainly fascinating aspects of your research that I've very curious about, including when different genes might be getting desilenced, actually, like in the cartilage with age. Some of those things that might be inhibiting hair growth or begin to inhibit hair growth. It seems like there's a lot of desilencing events with age that seem to happen that I don't know how they would play into this.
Okay.
Anyhow, this is,
yeah, sure. We'll be glad to chat. Thank you.
Yeah, I mean, that you've really just the level with which you've delved into the system is very impressive, Max.
Thank you so much. Yeah.
Yeah, and I'm sorry, I
like
apologize that we don't have you should be overwhelmed with questions, and normally you would be. And you know, I wish that we were in person so that we could all go to lunch and take time to hang out with you. But I will be contacting you as well, Max. And
okay.
well, do any of you have any questions before Max? Okay. Well, Max, amazing talk. I also loved your first slide, by the way. Was that actually digitally rendered, or was that a micrograph that you several micrographs?
Yes. So this is actually fluorescently labeled epithelial cells using H2B GFP, keratin 14 H2B GFP. It's a transgenic mouse that was produced in Elaine Fuchs's lab that we acquired.
Whose lab?
That's Elaine Fuchs's lab.
Huh. I
Rockefeller.
her sister at University of North Texas, Janet Fuchs, I think.
Okay. Yeah. And then so this was also following tissue clearing. So this was like a Z-stack acquired on cleared skin. So
okay.
Okay.
Yeah, because it's a huge field of view too, it seems like.
Yeah. So
that's why I was like, that was what was impressive to me. I was like, that is an incredible resolution in a large field of view. So that's, however you got that, and you took that, or you said it was?
Actually, it's my former postdoc. He's now in Yonsei University in Seoul, and he started a company, and his company makes tissue clearing kit. So he was very motivated to apply it to skin because of a very collagenous nature of skin. It's one of the toughest organs to perform tissue clearing. So if you take a brain or liver, it's much easier to tissue clear a brain or liver for imaging, but skin is notoriously difficult. So they actually had to optimize the standard kit to skin. I think, but they managed to do it, and it looked pretty cool.
Yeah, it's a really amazing work, Max. It makes me feel inadequate. So thanks.
Right. No, well, thank you. Very
well, no, you've done great, man. This is it's important. I have a lot of questions, but I will email them to you. And maybe next time in UC Irvine, we'll have a coffee or beer or something.
Sounds good. Yeah, thank you so much, Alex.
All right,
Thanks.
thank you. This was amazing. You did such a good job. Thanks, Max.
Thank you. Thank you. See you around. We will
See ya.
wait for your email. Okay, bye.
All right, bye.